interactions and models of phytoplankton growth.
Great. Thank you, Andrew. And I'm really excited to be here as part of this really fascinating session that I think is extremely important. So I'm hoping to add a small part to this discussion. So I'm decidedly not a modeler, but I'm really interested in understanding better what the micronutrient requirements are in phytoplankton and how we can better understand those to improve our projections of what's going to happen in terms of primary productivity in a future ocean. And the way that my lab does this is by using mass spectrometry-based proteomics, and you heard Richard discuss some of that work. And I'll go on and discuss some of that work both in the lab and in the field today. And what I'm really interested in is understanding where interactions between multiple nutrients are important in the ocean and how we can understand the mechanisms behind those interactions so that we can better parameterize these models that we've been discussing. So today I'll highlight some new work that we've been doing on manganese, as well as some work that I've been doing for a while now on vitamin B12, as well as interactions with iron. So we've heard about some important advancements in incorporating changes in phytoplankton physiology into these global models, but still the vast majority of global marine ecosystem models parameterize phytoplankton growth in a manner like this, where we've got growth rates being a proportion or being related to maximal growth rates, a function of temperature, a function of limiting nutrients, and a function of light, with little room for interaction amongst these. And most of them incorporate a minimum function of a limiting nutrient. So the idea is that there is one nutrient limiting at a time, and a very few of these incorporate, as Richard was mentioning, a multiplicative function of co-limitation. Okay, so we have a pretty limited way of incorporating co-limitation or multiple nutrient interactions into phytoplankton growth models at the moment. So as I said, most model conceptions of phytoplankton physiology do not meaningfully incorporate co-limitation by multiple nutrients or interactions between multiple nutrients. And so what I want to talk about today is briefly why not, and I'll give you a little bit of evidence then about why they should. I'll talk briefly about how we might be able to proceed, and then I'll use an example from the work in my own lab, giving us a window into why we might want to use a proteomic approach or a metaproteomic approach to try and do such a thing. Okay, so why not? Co-limitation is many things with many possible mechanisms, and this makes it inherently difficult to incorporate into these models for the reasons that Richard was mentioning and others as well. But to get us all on the same page, I'll describe here a few of the conceptions of co-limitation that have been put forward in the literature and that I use to frame the work in my lab. So co-limitation might be defined as multiple nutrients having simultaneously been drawn down to levels where the addition of both nutrients, either together or interactively, can stimulate the growth of plankton. And there's different scales on which we can consider co-limitation. So we should consider co-limitation at the level of a cell, of a culture, of a specific plankton group, or of a community. And these can be in three or at least three different conceptions. So independent, this is where you're trying to make a cake. You're out of both flour and sugar. No matter how much flour you add, until you get some more sugar, you're not going to make a cake. That's independent limitation. Then we can have instances of dependent limitation where one nutrient is required in order to access another. So instances of that include iron or zinc required to access organic phosphorus. And then we can have instances where there is substitution between different micronutrients. And these can be cases such as the substitution of cobalt, cadmium, or zinc in specific carbonic in specific enzymes, including carbonic anhydrase. So these kinds of nutrient limitation can manifest themselves at any of these levels, I would say. And then when we think about a community on top of that conception of individual cells, cultures, or phytoplankton groups, we also need to think about the fact that different community members can be limited by different things. We also need to think about the idea that community interactions can be a source of limiting nutrients, for example, vitamins, and that source might be limited by the presence of another nutrient. So when we start considering limitation of a community, we have added complexity here that can either be, in some cases, easily incorporated into models, or in other cases, much more difficult to incorporate into models as we know them now. So as I've said, co-limitation is many things with many mechanisms. So we can go from individual enzyme-based, so substituting a metal in a specific metalloenzyme, all the way through to black queen type interactions that we heard about yesterday, where we've got this public good of vitamin B12 that's being produced and consumed, and it's difficult to conceptualize. Another reason why not is that we're data poor. So Richard described some of the really important experiments that he did and others did that give us the important data to conceptualize these original models. Imagine adding all these interactions onto those types of models. The informative experiments are difficult to conduct, and it's not obvious which kinds of co-limitation are being observed and what the mechanisms behind those are, even when we observe them in the field. So, given all these difficulties, why should we incorporate co-limitation into these conceptual models of phytoplankton physiology in our global ecosystem models? Well, I'll advocate that there's lots of evidence now that these are really important in the field. And I'll just highlight one example that I was peripherally involved with. This is from a recent paper from Guillemard, Tom Browning, and Eric Achterberg's group, where they conducted a series of bottle incubation bioassay experiments in the South Atlantic around the Benguela upwelling region. And what they did was they did this full factorial matrix, Nalgene advertisement film, where they've added nitrogen, iron, and cobalt in a full factorial matrix, incubated the in situ communities for 48 hours, and measured the chlorophyll responses upon these multiple nutrient additions. And so you can see here the results of these experiments where it's blue, there's single nutrient limitation by nitrogen, and where you've got these outlines here, you've got co-limitation scenarios that I'll explain in a moment. So here's our single nutrient limitation. This is chlorophyll here on this axis here, and our control. When we add cobalt and iron, nothing happens. When we add nitrogen, you can see primary nitrogen limitation here. And then that was at experiment 4. In experiment 10, you can see shown here, we've got a scenario where only when we add nitrogen and iron together do we get a response of chlorophyll production in this community. So this is a clear instance where we've got this co-limitation situation with nitrogen and iron. Now if we move on to consider what happens in these instances where we add either cobalt or vitamin B12, you see a further increase. After you add nitrogen and iron, you see a serial limitation by either cobalt or vitamin B12 in this instance. And this was not isolated. You can see this in multiple examples from this field experiment here. So what this work suggested to us, we've got various lines of evidence that it's the B12 compound itself that was limiting the community, secondarily limiting the community here. And various other work that went into this paper demonstrated that the iron and nitrogen co-limitation scenario here was extremely widespread, but we were not able to make any conclusions about the nature and the widespread nature or how important cobalamin limitation or vitamin B12 limitation is because of a lack of mechanistic understanding of what's controlling this nutrient limitation in the ocean. So I'll suggest that this work demonstrates that co-limitation is widespread and important and highlights the fact that we don't have a broad enough mechanistic understanding of what's controlling these interactions in order to fully incorporate this result into geochemical models. So before I move on to some work from my own lab, I want to highlight another point here that other kinds of interactions matter. I want to suggest that micronutrient availability can impact plankton responses to other variables that are important for these conceptualizations here, including macronutrient utilization and responses to temperature that may be important for predictive models. And I've just highlighted a few examples here for us to be aware of. One is a classic that's been successfully modeled in some older work here. And then I've got some other examples here that I hope will become classics. That high vitamin B12 availability increases thermal tolerance in some phytoplankton, and that manganese and iron interactions increase resilience to variable light and iron conditions in some organisms as well. So I just want to highlight that maybe we're not just concerned with growth rate, there are other important interactions that can lead to variable stoichiometry and responses to other important conditions in the environment that we might want to consider. Okay. So I've explained why not. I've explained why we should. And I want to highlight now some examples from my lab about how we might think about proceeding. And I'm going to use field-based proteomic approaches to do that. Okay, so I'm going to share with you some data from a field campaign that I was lucky enough to get to participate in at the very end of my postdoc in Andy Allen's lab. I brought these samples back to Dalhousie University with me, where my lab group is now working on them. And what we did was we went to the sea ice edge in McMurdo Sound once a week for 4 weeks. And this was a project in collaboration with Debbie Brunk, who's here, and Dave Hudgens. And we took large volume samples once a week for 4 weeks. And the question that I'm asking with those samples from the water column is, what are the nutrient dynamics that are underpinning the productivity in this region? And the way that I'm asking that question is by using metaproteomic analysis via mass spectrometry. And I'm interpreting those using some bottle incubation bioassays that we did in conjunction with this, as well as some culture experiments to help us inform our interpretation. So we've got a short little time series here, 1 sample a week at roughly the same location at the sea ice edge. You can see it was a terrible place to work. So we took our samples for proteomics and a variety of other experiments as well. And then we also did these small volume bottle incubation bioassays. And so on this December date here, you can see our control yielded the same response as our iron, vitamin, as well as manganese additions here, suggesting that our phytoplankton community was replete for all these nutrients that we were examining. But as we moved further along in the season, we observed, interestingly, response when we added B12, when we added iron, and when we added iron and B12 together, as well when we added manganese alone, iron, and manganese and iron together. And I'm still pinching myself, or I'm still kicking myself for not doing the full factorial here, Phil, because I don't know what happened when I added all 3 together. But that's where the proteomics comes in, and we can try and just decipher what's happening in this situation. So I do want to highlight that we've got some drastic community composition changes happening here. We've got our phascis-dominated community here, which is a Promisia eukaryotic phytoplankton, and that we move into a community that was dominated by diatoms and dinoflagellates. So we've moved to our metaproteomic analysis via mass spectrometry, and the questions that we're asking with this work are, what are the major functional changes associated with this transition? Are there group or species or strain differences in the stresses that they're experiencing for these micronutrients? And can we decipher any important interactions between the micronutrients that might be at play here? And another question that I'm really interested in that I don't have time to talk about today is, what roles do interactions between phytoplankton and bacteria play in these dynamics? So this is a very quick overview, a quantitative proteomic overview of the functional changes that we saw comparing this date and this date here. So week 1 and week 3, and we've got the change in the proteome and the protein expression here, and the overall expression of that functional group. So I'm not asking any questions about who's there with this plot here. I'm strictly asking, what are the proteomic functions that are being expressed, which are the ones that are highly expressed, and which ones are expressed differently between these 2 time points. And I don't have time to go into this, but we have some interesting differences that we can interpret based on data that Richard and other groups have gathered that help us understand the responses of these chlorophyll-binding proteins and other important highly expressed proteins that we're interested in if we're going to think about changing stoichiometry in these plankton communities. So I'm happy to talk more about this, as well as the methods that go into generating a figure like this. My PhD student, Scott McCain, is responsible for the data analysis that went into creating this figure, and I'm really excited about the work that he's done to get this ready. But for now, I want to switch gears and ask questions more specifically about the micronutrient functions in the environment here. And to do that, I'm going to ask questions about specific functional groups. So first, I'm going to ask questions about diatoms. So can we pull out diatom sequences from this protein, or diatom-based mass spectra from this analysis? and try and understand their micronutrient physiology based on the protein expression patterns. And in order to do this, we need to turn to culture work, and this is some work that I did while I was a PhD student in Max Saito's lab, and that we've reproduced with other organisms in my own lab now. I'm showing you here the concentration of proteins that replace B12 requirements and that are involved in acquiring B12. And you can see that at high B12 concentrations, these proteins are expressed at low levels, and then at low B12 concentrations, these proteins are much more highly expressed. So what we can do now is go to our field measurements and observe the concentrations of these proteins with respect to total diatom contributions to the community and get a sense for the diatom nutritional status with respect to B12. And so here's the data. I'm showing you here the abundance of diatom MET-E protein in these gray triangles here relative to total diatom protein that we approximate in this case with actin. And then I'm showing you here the expression of the B12 uptake protein, or the protein we think is involved with B12 uptake relative to total diatom peptide contributions. And what you can see is across this time series, we go from what we expect to be low cobalamin stress to high cobalamin stress in these diatoms. And this is roughly consistent with those small bottle incubation studies that we were able to conduct. And so now we can look at these same organisms and ask about their iron nutritional status. And to do that, we're looking at the protein expression profiles now in these gray triangles here of a protein that's known to be involved in high affinity iron acquisition in diatoms. And what we can see is a roughly similar pattern where we've got over time increasing expression of this protein that suggests to us that these diatoms are experiencing stress due to the lack of iron. So we've got high expression here. So what we can see is that there's quantitative proteomic evidence of a transition into simultaneous iron and cobalamin stress in these diatoms in this community. Now, because we know that protein-based information can tell us more than just, is this from a diatom? It can tell us which kind of diatom these peptides might be generated by. We can ask questions about which diatoms are experiencing these micronutrient limitation scenarios or micronutrient stress scenarios. And what we can see, I'll just summarize this quickly, is that we think that Fragilariopsis in this community is experiencing simultaneous cobalamin and iron stress. So in the interest of time, I'll move on to talking about Phytopl and manganese, but if anyone is interested in talking about community interactions, I'd be very happy to talk for hours about that. So now I want to tell you about some work that we're trying to do to decipher whether we can see signatures of manganese and iron stress in Phytopl communities across this transition here. And in order to do that, there wasn't very much culture-based evidence to go on, so my lab had to do some experiments looking at the protein expression profiles in Phytopl in the lab in response to simultaneous iron and manganese deprivation. I'm showing you here the response, Fe over FM response in our 4 conditions, and chlorophyll per cell here, suggesting that there's a change in the photosynthetic responses of Phytopl to these 4 different conditions. And we asked what we could see in the protein expression profiles here. I'm over time, or I'm almost over time, so I won't go into this, but I want to highlight that we see major changes to the photosynthetic apparatus based on iron availability, some finer scale changes based on manganese availability, and we see really interesting differences in flavodoxin and plasticyanin responses to iron and manganese that help us identify these as 2 potential proteins to track in phytoplankton communities that might tell us something when we measure them together about iron and manganese. We have more work to do to translate this culture work into the field, but just a preliminary look at this. I'm showing you here on a different kind of plot the expression of plasticyanin and flavodoxin across different treatments, highlighting the low iron and the combined low iron and low manganese. And what you can see here represented in the field data is hints that this protein expression roughly mimics that of that protein expression profile under combined low iron and low manganese. Okay, so I'll just sum up here and suggest to you that I hope I've shown you that proteomic measurements can help us document and explain the molecular and ecological bases for columation of primary producers in the ocean. I showed you that we require field and experiment data to fully interpret those metaproteomic measurements, but that it has a potential to resolve taxon-specific responses in situ, and it has the potential to identify key in situ interactions and mechanisms that might be important for capturing in models. So I'd like to thank my lab group and then all the great collaborators that have helped me get this work off the ground, and thank you for your attention.
That was great.
Thanks.
I really like how the proteomics are really starting to help us understand the nature of the response. But I had a question on the Browning results.
Yeah, sure.
It seemed from face value that you could explain those as a sequential onset of Liebig limitations. And is that a correct impression, or did I miss something more?
No, I think, well, in terms of the nitrogen and iron, I think you're right that based on the limited results that we have, we could, Tom and the group could capture most of the responses that we saw with a Liebig-based model. And so that was encouraging in some ways, but then if we dug a little bit deeper, especially when we consider the cobalt and B12 interactions, we could not predict based on Liebig, and we left that open there.
Yeah, I think trying to bring out those departures from Liebig would be great, because that's where our current models would
absolutely
assuredly fail.
So I think we have some more work to do with approaches like this and others to tease that apart. But yeah, thank you for bringing that up.
Hi, Erin. Thanks. That was a wonderful talk. I have a question. Have you gotten to the point with this where you can sort of figure out if it's the same organisms that are showing multiple limitation at the same time, or like with certain things like cobalamin and B12, there's shifts that can happen. But are you
it's a great
there
question.
Yet?
So I think I can confidently say that Fragilareopsis is experiencing cobalamin and iron stress at the same time, but whether it's the same Fragilareopsis, I don't know what we mean by the same Fragilareopsis anyway. but so I think that's an important question, and in the field, where does the transition from a functional group to a specific I don't know where that line is. It's just the same question about what is a species, what is the important level to consider here. I can't exactly answer that yet, but I think we have the chance to get closer the more data analysis that we can do with this. So yeah, thanks for that great question, Drew.
Hi, Erin. Thanks for the talk. I was wondering if you're considering the speciation of the metals that you're adding, because I know for iron, a lot of it might be iron 3. They might be using it to acquire a ligand, and have you played with that at all?
Yeah, absolutely. I think there's a lot of potential for important work to be done there, and we didn't look at that at all in this study. I know there's some important work that Drew and Kristen and others are doing that can start to speak to that. So coupling the approaches that they've been doing with our field proteomic analyses would be great. So the responses that they're seeing, how can we combine that? So that's a great question. We can't answer that right now from this data, but yeah, there's hope for. Thanks.
I'd like to thank Erin and push on to the next talk.